So this is a cardiac catheterization, which is a series of live images that we take via X-ray. And you can see the heart's beating. You can see a catheter all the way on the left injecting contrast into the coronary tree. And right where that red dot is you can see a blockage. We can fix that by opening up that blockage and putting in a series of stents to restore blood flow to the heart. I like showing this case because although I took care of this patient, my wife, Dr. Abraham, actually performed this procedure. Although we can restore blood flow, the heart itself still gets injured during all the time that it's blocked. If that injury is severe enough, you get a condition called heart failure. So this is an echocardiogram of a normal human heart. You can see that all 4 chambers are beating quite vigorously. If you get a heart attack and have severe enough injury, it looks more like this, where the chambers are fairly weak and unable to pump. This condition is called heart failure. So how do we treat heart failure? So I want you guys to do a little bit of a thought experiment with me. Imagine that you're driving your car and you hit a pothole, and all of a sudden your engine isn't working so well and it's making a funny noise and your car won't go. So you take it to the mechanic. Imagine what you would think if the mechanic told you this. From now on, only one person in the car. You can't have any passengers. It's going to stress the car out too much. From now on, you can't take it above 30 because it's going to stress the car too much. And from now on, no going uphill or into the mountains because it's going to stress the car too much. So I think in this circumstance, any of us would ask, well, why can't you just fix the engine? So this is how we treat heart failure. We reduce the stress on the heart. We can do this in a variety of ways, by reducing the amount of blood the heart has to pump, by reducing how fast it can go, or how hard it has to beat. All of these therapies are incredibly effective, but they don't get at the problem with the engine. And in heart failure, one of the most common dysfunctions are within the mitochondria. So what are mitochondria? Mitochondria are the part of every cell that make the fuel. They have 2 membranes, and embedded in this inner membrane is a series of proteins that turns everything you eat into fuel the heart can use, a molecule known as ATP. In heart failure, mitochondria don't make enough fuel. We've chosen to study this form of dysfunction in a series of rare diseases known as the mitochondrial cardiomyopathies. And cardiomyopathy is just a fancy way of saying heart failure. These are diseases that occur primarily in children and in infants who are born with mutations in their mitochondrial proteins. And although the mutations can occur in a wide range of proteins, these children will often get a cardiomyopathy or heart failure. So in order to understand our approach to study these disorders, I thought I'd take a little detour and tell you about my background. So although I was born in India, I grew up in a small town in Venezuela along the northern coast. And I had never done research or been in a lab or done any kind of science until the sophomore year of college, where a friend told me to check out some labs. And I happened to meet Andy Bass. Andy turned out to be a wonderful mentor. And what Andy does is he's a neurobiologist who studies these fish, which are known as the plainfin midshipmen, found along the northwestern coast of the United States. One of the interesting things about these fish is that the male, who's in the middle there, sings to attract the female to its nest. And so Andy was showing me the lab when I first met him, and we happened to go into a room where a student was performing an experiment, and I heard this. So that humming that you hear is the song of the male, and all the clicks are electrical activity in the brain of a female who's listening to that song. And, you know, for me, that was it. I was hooked. I thought this was the coolest thing I'd ever seen. And since then, I've kind of dedicated my career towards understanding how electrical activity affects cellular function. So following that experience, I went on to grad school, and I happened to have the privilege of working with David Yu, who unfortunately passed away about 10 years ago. And I learned a lot of things in David's lab. I want to tell you about 2 of them. So if you look at this picture, I'm not sure exactly when this picture was taken. It might have been 2002, it might have been 2003. But one thing I can tell you is that this picture was taken around 9 o'clock at night. And that's because David and I had finally, after several hours of battling the electrical equipment you see behind us, reduced the noise in it enough to get some really nice recordings, those green squiggles that you can see on the screen. And if you look at me, I'm pretty tired. I want to go home. But David has just boundless enthusiasm. And I think that was one of the things he taught me, that a lot of what we do ends in failure, so you got to celebrate even the smallest of victories. The other thing I learned in his lab was calcium channels. So these are proteins that are embedded in the membrane, and whether it's on the surface of the cell or within the cell, and what they do is they take calcium from one side of the membrane to the other. I went on to study these within the mitochondria during my postdoc with David Clapham. And so since that time, we've chosen to study the mitochondrial calcium channels. The one we study in particular is this one known as the mitochondrial calcium uniporter. So what does calcium do within the mitochondria? So I'm going to use exercise as an example. So whenever you exercise, calcium levels go up within the cells in your heart, and they get transmitted to within the mitochondria via this channel that we study. Within the mitochondria, calcium acts as a sort of accelerator. It boosts the activity of these proteins to generate more ATP. This is what allows you to exercise and run hard. So in these diseases that we study, there are mutations that affect the ability of the heart to make ATP. That's that cellular fuel that the heart uses. They're not making enough ATP. What we've spent the last few years defining is that there is a feedback pathway that the mitochondria uses. It senses this reduction in ATP and actually makes more channels. These channels then let in a lot more calcium, and that calcium can then boost the activity of the proteins that are there to restore or even rescue this energetic failure. That's so mitochondria engage this pathway to preserve energy production in heart failure. So that work took several years, and it was led by Enrique Valderas, who's a postdoc in the lab, along with two other postdocs, Sal and David, as well as Sandra, who's a graduate student in the lab. And defining that pathway was really important, but it kind of gets to the deeper question, which is, can we actually use this channel as a potential therapy for mitochondrial disease? So to get at that question, I'm just going to show one proof of concept type of experiment that we did in collaboration with Eileen Rodin, who's a Drosophila geneticist and a nephrologist in our Department of Internal Medicine. John is a technician in our lab, and he does this experiment that I'm going to show you here. He's got this platform that's sitting in a bowl of water, and he's just going to dump a bunch of flies on there. And these flies are normal flies, and they're just going to take off within a few seconds. These flies have mitochondrial diseases, and so they are much weaker. Uh-oh. Let's see. Oh, there we go. Okay, so let's give him a second there. Yeah. So these flies are much weaker, and they can't take off. They have mitochondrial disease. So what we did was, let's see if we can vary the amount of calcium and actually affect the viability of these flies. So normal flies, it took them in about 5 seconds, half the flies were gone. If they have mitochondrial disease, it takes them closer to 10 seconds. If you actually get rid of that calcium channel, they're pretty severely impaired. Half of them can't even fly anymore. But now here's the key experiment. We actually boosted the levels of that channel, and what we found is that we could rescue their weakness and turn flies that had mitochondrial disease back into have the strength of flies that were otherwise healthy. And so since then, we've been trying to see if we can perform this same kind of boosting using drugs instead of genetic tools. And for that, it's a collaborative process in which we've enlisted the help of multiple different collaborators, including Hari Venkayapati at the Huntsman Cancer Institute, Erhuk Chow, and the Department of Biochemistry, and Randy Peterson from the College of Pharmacy. And this work's been funded by both the Pivot Center here at the U, as well as the NIH, and some private companies that have given us access to some of their compounds. So our goal ultimately is to use this channel to turn a heart that's beating weakly and ideally get it to accelerate and turn into a heart that's beating normally. So with that, I'd like to thank you for your attention, and these are the people who actually did all the work. And I'd like to give a special thanks to the Nora Eccles Treadwell Foundation, which really went out of their way to support a lot of this work. Thank you very much.